Hi, everybody. We're back from this short break, and we will continue with our second talk of today by Athina Eleftheraki, who is going to talk to us about post-transcriptional regulation of the timPR type 1 toxin-antitoxin system in the organism Salmonella. And with that, I'll stop sharing my screen.
Let's see. Do you?
Yes, we see your full screen and you. All right. Thank you.
Hello, everybody, and welcome. So, as Eva said, today I will talk to you about my main project, which is the post-transcriptional regulation of timPR type 1 system in Salmonella. I am a PhD student at Eric Holmqvist Group. My co-supervisor is Gerard Wagner, and I work at the Department of Cell and Molecular Biology at Uppsala University, and I'm also part of Uppsala Antibiotic Center. So first I will start with, So first I will start with a brief introduction about toxin-antitoxin systems. So the idea is that in principle, we consider them bipartite systems. One gene encodes for the antitoxin and the other gene encodes for the toxin. Under normal growth conditions, the antitoxin can act on the toxin and the bacteria can continue growth. However, under different stressors, the antitoxin can be either degraded, deactivated, and then the toxin is free. When the toxin is free, then it's a self-damaging toxin, and we can see toxicity upon overexpression. So there are two important key features for toxin-antitoxin systems. The first one is that the toxin is self-damaging, which means that it kills the host cell, the bacterium itself, and the antitoxin protects against the toxin activity. One other important feature that we need to remember is that the toxin is usually much more stable compared to the antitoxin. So toxin-toxin systems, they are widely spread. We have a lot of them, and we distinguish them in 8 different types based on the mode of action and the nature of the antitoxin. What is striking is that TA systems, we can find them everywhere. We can find them on chromosomes or bacterial plasmids, and we can find them in all bacteria. We can also, of course, find them in human pathogens. And here I will just give you two very brief examples of human pathogens. We can find about 79 TA systems in Mycobacterium tuberculosis. And also in research in Staphylococcus aureus, they found a lot of TA systems, but also great diversity in the type of TA systems. What we also need to keep in mind is that these numbers may actually become higher because we find many more TA systems regularly. So we know that they are a lot. We know that they are in all bacteria. But what are the biological functions of TA systems? So the biological functions are various and they are diverse. I will just briefly mention some of them, and I will start with phage defense. Some TA systems have been shown to act as phage defense elements. And what that really means is that upon phage infection, so once the phage can actually infect the bacterium, then these TA systems can be activated and induce self-killing of the bacterium. By self-killing the infected bacterium, then you can control the spread of the phage in the population. Another function that is actually relevant to antibiotic resistance is the plasmid maintenance function of TA systems. So some of them, especially when found in plasmids, they can maintain the plasmid in the bacterial population. So once you have a TA system and you lose the plasmid, now you need to remember that the antitoxin is less stable. The toxin that is more stable is going to stay in the bacterium and induce self-killing. One very relevant function as well is the biofilm formation. In this case, we have seen that some TA systems have a role in regulating the biofilm formation and the motility of the bacteria. And this is especially important when we consider the efficacy of antibiotic treatments in biofilms. And finally, some TA systems, especially type 2 TA systems and one type 1 TA system, they have been seen to have a role in bacterial persistence. And what happens here is that in a susceptible antibiotic population, you have stochastic expression of these TA systems, which leads to slower metabolic activity. When this subpopulation has lower metabolic activity, cannot be targeted by the majority of the antibiotics that act on rapidly growing bacteria. So once you retrieve the antibiotic treatment, this subpopulation can give rise to a new population, and then we have a new infection. So these are some of the various functions that TA systems can have. And the question is if we can use now TA systems in human health. There are a couple of ideas that have been suggested in the field, and I will start by one. So it has been suggested that TA systems themselves can be used as therapeutic targets. So if you remember, I mentioned two functions of TA systems. One was the bacterial persistence, and the other was the plasmid maintenance. So the idea is that by targeting TA systems, we can actually target the persistent bacteria, and then we can have a more effective antibiotic treatment and not relapsing infections. Second, when we have antibiotic resistance genes that are found in plasmids, if they co-localize with a TA system that actually has a pressure to keep the plasmid in the population, we can target this TA system, so the bacteria can lose the plasmid without self-killing. I also mentioned that some TA systems have been found to have a role in phage defense, and that is specifically important in cases that we use phage treatment as bacterial killing. And what we can do now is that by understanding these TA systems and the phages that they are actually induce resistance to, we can have a more effective phage therapy. And finally, we can use the TA systems as direct antibacterial strategy. If you remember, I told you at the beginning that upon overexpression, TA systems are toxic. And this is the function that has been suggested to be used as antibacterial strategy. Induce expression of the systems, kill the bacteria. So I talked about different functions, different TA systems. They are everywhere, and as you can already imagine, they are different. So if we want to use them, we first need to understand them. And this is where I will focus today. I will focus on one specific TA system, which is called timPR. This system belongs to type 1 TA systems, and the timP, and the timP encodes for a small toxic membrane, while timR encodes for a small RNA that can bind on the timP mRNA by base pairing, inhibiting its translation. So what we have seen before, when we first discovered the timP-TMR system in Salmonella, was that upon overexpression of timP alone, we can see bacterial growth inhibition. However, when we co-express TMR, we can rescue this effect. What we have also seen previously is that as a proof of principle, timP can induce persister formation upon treatment with ciprofloxacin. But the question is still there. How is timP-TMR system working? If we try to see it as a linear model, what we see is that timP, in the absence of TMR, can actually be translated, and then we have growth inhibition. However, in the presence of TMR, the small RNA is binding on the 5' untranslated region of timP mRNA, inhibiting its translation. What is striking, if we see it as a linear model, is that TMR binds upstream of the Sindel-Gardner region. So in bacteria, the translation initiates at the ribosome binding site, which includes the Sindel-Gardner region and the start codon of the open reading frame. So a canonical mechanism of small RNA regulation inhibition in bacteria is that the small RNA binds in close proximity, if not on the Sindel-Gardner and the start codon. So it was very striking for us to see that the small RNA actually binds far upstream of what we would expect to see. So the first questions already raised, and these were, how can translation initiation occur on timP? Is it actually occurring on this site, as we would expect? And how can timR inhibit translation since it binds so far upstream of a canonical translation initiation site? To answer these questions, we started by looking at the 2D structure of the 5' UTR of timP. So what you see here is only the 5' UTR, and we have some key findings. The first one is that timP is has a well-structured 5' UTR consisting of 4 stem loops. You can see here that timR binds on stem loop 2, which is still far upstream of the Sindel-Gardner sequence, which is now well sequestered on SL4, making that an unfavorable structure for translation to start with. Oops, sorry. Yes. So we wondered what are the important features of this 5' UTR, which is quite long, for timP translation. We started by making some truncated versions of timP, and the idea is that we would truncate SL1, SL1, SL2, and everything upstream of the Sindel-Gardner region. So you can see that already when we lose SL1, we see no translation of timP. And then we wanted to make some deletions further on to be able to narrow down where is the important features for timP translation. So first we deleted the upper part of SL1. And here, again, you see that there is no translation for timP. Then we deleted only SL2, and in this case there is no effect on translation of timP, indicating that this region is not responsible for translation initiation. It seems to be mainly responsible for timR binding. And finally we truncated SL3 and we lose again translation. So that was a first indication that probably this part of SL1 and SL3 are responsible for translation of timP. And of course, we wondered if there is an interaction between these two stem loops. To answer that, we first went to check if we can see evolutionary conservation of this interaction among enterobacterial species. So this interaction that we predicted is here. It's SL1 and the loop of SL3. And what we see is that this interaction is well conserved among their bacteria, indicating that this interaction is probably true. But of course, we had to test it. So what we did is that we induced 2 mutations. We have the M2 mutation, and this mutation would disrupt the pseudoknot formation from the SL1 site, and M2' mutation that would disrupt the pseudoknot formation from SL3 site. So I figure that I may forgot to say that, but this interaction, we call it a pseudoknot. So what we see when we introduce the interactions is that the mutations is that we completely lose translation. And the translation of timP can be restored when we actually restore the pseudoknot formation. So that was a great indication that this pseudoknot formation is essential for timP translation. We also tested if timR can now inhibit translation of timP when we restore this interaction, because we actually saw higher levels of timP translation, And we saw that timR can still inhibit all the molecules that are going to be translated. We saw what was interesting is that with this mutation, we saw higher levels of translation, but we could still inhibit fully timP. So we wondered if we have more than one structures that can be that are found in the mixture and can be translatable or non-translatable. So we worked around this idea, and now I show you some different experiments. Here, the first experiment is called electromobility shift assay. And the idea is that we have radioactively labeled timP mRNA, which we incubate with non-labeled timR small RNA, and we look at the binding rate of this interaction. So these are time points. And what we see is first that we cannot fully bind timP with timR, but we can see that in 10 seconds, which is pretty fast, we already see some binding of timR on timP. When quantifying 3 independent experiments, what we saw is this is the binding curve, and what we saw is that we could easily distinguish 2 different rates, one very fast binding rate and one slower binding rate. However, in the same conditions as we do the EMSAs, where we cannot fully bind all timP molecules, we can fully inhibit translation of timP. So now, having that, and having the idea that we cannot fully bind timP, and also having the 2 different rates, we consider that maybe there are more than one structures and cannot be fully bound of timP of timR if not translatable. So to answer that, we did again an EMSA. Now we have radioactively labeled timR and we have called timP. So we check the binding with wild-type timP and we can see a similar pattern as before. We check the binding with a timP mutant that cannot form the pseudoknot, and what we see here is much less binding compared to wild-type. And finally, we check the binding with the compensatory mutant, and we see again a binding of timR. So when we also quantified this, that was striking because what we saw is that when we don't have the pseudoknot, actually we have much less binding of timR on timP, while when we compensate and we probably have more pseudoknoted structures in the mixture, we also have more binding of timR on timP. So maybe here, just to summarize a bit what we had seen up to now, we had pretty good indications that we have more than one structures. We have a translatable and a non-translatable structure. And we also have pretty good indications that timR preferentially bind on the translatable structures, so it can inhibit translation of timP. If timR binds on the translatable structures, what happens with the pseudoknot formation that we know that is essential for timP translation? To answer that, we went to structure probing of our compensatory mutant, so we have the pseudoknot, in the presence of timR. So it's a simple experiment in a sense. So what we have is radioactively labeled timP, which we incubate with timR so they bind. And then we cleave with lead acetate, which is going to cleave all the single-stranded nucleotides, or with T1, which is going to cleave all the single-stranded Gs. And what we see at the end is a gel like that, where everything that is cleaved is going to be single-stranded. So we will focus only on 2 regions. The first region that we focus is the region where timR is binding, which is here. what we see is that upon timR binding, these are increasing concentrations of timR. So before, when there is no timR, we can see that T63G is not protected, which means that is cleaved. However, in increasing concentrations of timR, we can see higher protection of the G from T1 cleavage because timR is binding. Similarly for G76. On the other side, for G79 and G81, what we see is that in the absence of timR, this region is protected from cleavage because it's part of stem loop 2. However, once timR is binding, this region is less protected because now timR binds here, so this region is less structured, is less protected for cleavage. What we were actually looking for, though, was to see if we can see protection or loss of protection on the pseudonode. So we went on SL3 side of the pseudonode, and what we saw is that with the increasing concentration of timR, we can actually see less protection in this area, indicating that this area now is losing the pairing and is becoming single-stranded and available for cleavage. The final question, though, was if timR, okay, we know that timR binds preferentially the pseudonoded structures, and it seems that it opens up the pseudonode, so the translationization cannot occur. But is timR inhibiting only the pseudonode-dependent translation of timP? So to answer that, we actually could not work with the mutants that do not carry the pseudonode because there is no translation. So what we did is that we exchanged the Sindel-Garno region of timP, which is well sequestered in SL4, with a more open Sindel-Garno, which is more accessible to 30S binding. And what we saw is that when we have a wild type situation, the difference is of course the Sindel-Garno. timR can actually inhibit translation, but not fully, because now we have a Sindel-Garno that is accessible for 30S binding. When we don't have the pseudonode, there is no effect on translation. And when we restore the pseudonode, we see the same effect as the wild type. So that was a very good indication that timR is actually inhibiting the pseudonode-dependent translation of timP. To summarize the findings, the model that we suggested was that upon transcription of timP, we have a pool of translationally inactive structures with a well-sequestered Sindel-Garno sequence, SL1, SL2, SL3, no interaction. However, upon slow structural rearrangement, we have the formation of the pseudonode between SL1 and SL3, which could facilitate 30S or S1 ribosomal subunit binding on the pseudonode formation and then move forward to the Sindel-Garno region and initiate translation. However, in the presence of timR, timR seems to be disrupting this pseudonode formation and inhibiting translation of timP. So with that, I would like to thank Holmquist Group and Gerard Wagner for their for their support, especially Eric, that we co-authored the publication. Uppsala Antibiotic Center, it's very nice to be part of Uppsala Antibiotic Center, and all of you for listening.
Thank you so much, Athena. Thank you. This kind of talk puts me back to my bacterial genetics years of studying, how the different regulations used to work. Very cool work. Let's see if anybody has any questions for you. Meanwhile, maybe they think about it. You guys can put the hand up or you can write the question in the chat. Um, I want to ask you, for those that are not familiar with how you actually work in the lab with this, because you were presenting to us, all these different changes that you make to the sequences, and then you can assess, how the bindings are working or if it works or not. How do you actually make these changes happen?
The mutations.
Yes.
Okay, let's see how many technical terms I can use or not. So the idea is that we know the sequence of the gene
Mm-hmm.
that we know, and then we use PCR, polymerase chain reaction, and the idea is that we amplify this gene, but in this case, we use a different um, how can I say now? We use primers that actually carry the mutation, which
Mm-hmm.
means that once we start amplifying, every new amplification process will carry the mutation of interest.
Mm-hmm. Mm-hmm.
Once we have that, then we can use that, which is in our case a lot of times part of a plasmid,
Mm-hmm.
and we can use this mutated plasmid and put it in Salmonella.
All right. So you include, so you have this team R that you want to change, for example, in a plasmid, and then you replicate that plasmid,
In
including the change
Salmonella.
in
Yeah,
Salmonella.
so we do the PCR,
Uh-huh.
right?
Mm-hmm.
And there you have, of course, you have a great amount
Yeah.
of,
Mm-hmm.
of your product, and then we store the plasmid in E. coli. We use top 10 competent cells because it's much easier, and then we transfer and transform this plasmid into Salmonella.
Mm-hmm. And once it's in Salmonella is when you do all your experiments with that new mutated one.
All the in vivo ones, yes. If we want to work in vitro, it's a bit different because there we need to use this mutated gene as a template for transcription, in vitro transcription. And then we work with the mRNA that comes out from the in vitro transcription.
All right. But those changes that you make are like PCR based. That's how you introduce the changes, either to the one that you express from the plasmid or the one that you then put for the in vivo into the Salmonella ones. All right. So where are you going forward with this? What are your next steps in this project?
So we have performed a big analysis on the gene, and I can say as a teaser that we have identified the more detailed post-transcriptional regulation on TMP, which seems to be quite interesting, I would say. This is the idea for now, at least, because me as well, I'm going to finish in a year or something.
Mhm.
But what we're also working with is to see if we can see the 3D structure
Uh-huh. Of that pseudoknot on the team R and
of
how.
TMP and how TMR can change that. Yeah, but this is a bit more challenging, I would say.
I see. I see. So 1 year left
Mm-hmm.
for this thing. Very nice, very interesting. Anybody out there? I see both your supervisor and co-supervisor are here as well. Thank you, Eric and Gerhard, for joining us today. Some of the other PhD students as well, the new PhD students. Really good, Athena. Very happy to see how the progress is going. This very in detail molecular biology of things. Sometimes, when you work in AMR and you know about the aspects of, resistance and gene expression, you don't really think about gene expression so much, right? But as you are saying, and you were putting it in context at the beginning of your talk, in order to be able to use a tool, we need to really understand how it works first, right? And that's where your project and where your work falls into the very bigger picture, which is these systems seem to be everywhere, what are they doing, how do they work, and then can we actually apply it for something else. It reminds me a little bit, on about CRISPR-Cas, if we put it into a bigger context of something that maybe someone have heard about regulation of expression, that CRISPR-Cas is something that was in the bacteria just to protect them. And by really understanding the mechanism behind, then it was able to be made into an actual tool that can be used in molecular biology. So it's really cool. Thank you so much, Athena.
Thank you.
And if nobody else there have any questions, then we are going to conclude today's webinar. We welcome officially the summer break at the activities at the UAC. And we hope to see you back at the end of the summer. In September, we will have our first webinar and some other new things that will be coming up in the spring as well. And also, just to tell you guys that the Uppsala Antibiotic Days will still be happening in 2026 in June 15th and 16th. So you can already save the date for that big and really cool conference that will be happening in Uppsala with this interdisciplinarity focus on AMR. With that, I thank you all for your attention today, your presence, and see you in the next event.